look at problem number 1 here. It says DNA samples taken from rats. This is a released question, by the way. This would be a computer question, so you got to expect. You can go online at the OST, and I'll put the link in this review when I post it on Schoology. But you can go online and actually do this, like choose which one it goes with which one. They'll literally ask you to pick it up and put it in the appropriate space. The DNA samples taken from rats from the same colony produce 4 gel electrophoresis bands. Remember we talked about in the earlier explanation and in your class reading, gel electrophoresis is when you can take proteins or DNA and you put them in a well and then you run the well and you get a pattern. The pattern corresponds with similar proteins or similar DNA molecules. Similarity reflects a similarity in the DNA sequence, in the RNA sequence, or in the protein amino acid sequence. So depending on what you're running in the electrophoresis. So DNA samples taken from rats from the same colony produce 4 gel electrophoresis banding patterns shown. So you got RAM B, C, and D, and there's A. Determine the relationship between rat A and all the other 3 rats, B, C, and D. So one of these is an identical sibling, another one's a sibling or a parent, and the third is completely unrelated. Now, to be honest, even though the details are complicated, the reality is pretty simple. Which one of these 3 matches most closely? Which one of these 3 relationships is the closest relationship? So the closest relationship is going to have the most closely related pattern, and the one that's unrelated is going to have the furthest pattern away, and the one that's in the middle will be in the middle. So place the relationship in the labels of blank boxes under the electrophoresis patterns for rats B, C, and D. Place only one label in each blank box. You may use the label more than once. So where would you use, let's take a look at this. This particular rat A has a big pattern, 2 little ones, then 2 same widths, and a third one that's thinner in about this area. You'll see A and C have identical patterns. Identical. Well, I'd have to go with those 2 being identical. So identical would go here, right? I would think. Sibling. So a sibling is identical would be to rat A. The next, so that takes care of identical, that's moved, and I can use it, you place only one symbol in each box, but you could use the label more than once. Okay. Well, one would assume if they had 2 of them, they were all 3, this C and D had the same identical, they'd be triplets, right? In this case, they're just twins. Well, I look at A and I see that this is the same, and here there's only 1 band instead of 2, and that band is down here in this one rather than being up there. Then you have this band is the same, but then it's missing this thinner band, and there's a band that's a little thicker that's down lower, and then there's a third band that's thin here but thick here. So this to me seems like it's very similar. They're similar but not quite. How similar are they? Well, let's count them. This is 1 band that's the same. This is the same. I'm just going to mark the things that they're similar or the same. Here's one. One of the 2 there is similar to that one. And then this band here is still in the same place here. And then this band here is similar. It's in a similar place. It's thicker on the right than it is on the left. But to me, there's 1, 2, 3, 4 similarities between A and D. So there's 4 similarities. So at that point, I'm going to have to say it's similar enough that it could be a brother or parent. Sib or parent. And the esports team is practicing in the room, so you're going to hear talking. You can't ask them to be quiet. I mean, they are. It is a sporting event. All right, so that takes care of 2 of the 3. The next question is this rat B, is it a sibling or parent? We know it's not identical. Is it a sibling or parent, or is it unrelated? Well, how do I know it's not identical? Well, look at it. It's not identical to this. Now, this one is similar, but it's thinner. It's in a similar place, but it's thinner. Here, there's two thin ones. Here's a real thick one, and then this one's lower. It's not even in the same spot. It starts earlier, ends about in the middle of this. This one begins here, ends down here. Very different. Then this band here is much higher than this band is. This one is about at the same place. It's thinner, and this one's thicker. And this one is just as thin, but it's even in a different place. Is there any similarity between B and C? I don't see any similarity between B and A and B. There are four similarities between D and A. You can argue some of those, but there's at least a couple similarities between A and D to four similarities between A and D. A and C are identical, but A and B have no similarities as far as I can tell. There's not a single band that's the same. So I'm going to say no. I'm going to have to choose unrelated. And to get 1 point on this question, you have to pick up identical sibling, and it has to be put here. Sibling or parent has to be put here, and unrelated would have to be put there. You'd have to put all three in the right place to get 1 point for this question. So that's how tricky these can be. All right. And the answer's down here for the scoring guide. But that's the reasoning. That's how it works. Okay, so we're looking at this. We're looking at amino acid sequence differences, and they're going to look at differences, and that's key. And we're going to try to figure out which ones belong in each of these boxes. These two are the ones that have the least amount of difference, right? Which two on this chart have the least amount of difference? Well, B and C have only one difference between them. So these two have to go here. So B, if I put B here and C here, that's reasonable. They're shared only one difference. See? So B and C, now I don't have to worry about B or C. I just got to worry about D's already placed, right? So D's there already. And the question is, does A go here? Or does A go here? Where's that going to go? Well, how closely related is A to D? A and D have 11 differences. A and D have 11 differences. How closely related is let's see, which one's left? How closely related is B, C, A, B, C? Well, how closely related is E, which is the other difference, E to D? E and D share 3, so they're really closely related. Where A and D have 11 differences. So what if we placed E here? That means there's 3 differences between D and E, so that would be 3 differences. Just like there's only 1 difference between these two. But A has, that would mean A would have to go in the only slot a bit left to it, which would be here at the bottom. And you have to ask yourself, is that right? Does that show a lot of difference? How closely related is A to any of these? Well, A is 11 at the closest and 14 at the furthest away. So that makes sense that A is all the way out here. D and E share 3 differences. There are 3 differences between them, so they're very closely related. But then you look at D and E, and between D and B, let's say, between B and D, there's only 9 differences. That's a lot smaller than 14, like between A and E. So all this makes sense. This is the best arrangement that you can make using this one chart. The key here is that you're just looking to see, why is this tricky? Because normally we look at how many different common things they have when we did a cladogram. Here they threw you for a loop and they said how many of these are different. These are the differences. So the larger number is not an increase in similarity, it's actually an increase in differences. So they're going to be the ones that are furthest away. So A being different from B, C, D, and E, the most different from all of them, should be out here. B and C are very close to one another. We can see there's only one difference. They should be together. And D and E should be together. Notice they have a common ancestor right there. They only have three differences. So this setup here shows clearly that B and C have a common ancestor recently. D and E has a common ancestor relatively recently. Where A and the rest of them have a common ancestor way, way back in the evolutionary history. Okay, so let's take a look at this one. This is just, now they don't give you a chart. They just give you the actual similarities. Now, a lot of you are going to get confused when you look at this position. This is just a position where the DNA, it's either in the very first position or it's at the very end of the gene, right? These are the different taxa, right? Avian, meaning bird families, and these are these are different bird families. And here are the differences. The red are differences. So this is the first one that has T, but then these are different from T. Right? These are all C except this one, which is a T. These are all T's. So the black shows you that they're all where they all share in common. The red shows you where they're different. All right. So we have to we're questioning, they're giving us Vero. The Vero's here, and they're telling us, and forget about how they're pronounced. Forget about all that. Just look at it and look at the similarities and differences. And I think if you looked at it, you saw that these are all the same. I think it's clear that the black letters are, without anybody explaining it, I think I could look at this and say, well, these are all the same, and this is where they're different. At position 1, at position 4, position 5, there's only one difference here between them, etc. The question they're probably asking, we're going to read it in a minute, but the question they're probably going to ask me is to take one of these and place them where they belong here. So it says DNA sequences for a number of bird families in an order of passiformis are shown. Differences in DNA sequences between the Pitloris family and other bird families in this order are highlighted in red. And they even tell you don't have to figure it out, but again, just scan the diagram. What does it make sense? And then read and try it. It should take you a very short period of time. Then an incomplete phylogenetic tree that notes important differences in the DNA among the bird families is also shown. Use the DNA sequence data to complete the phylogenetic tree. This is the phylogenetic tree or cladogram, whatever you want to call it. Move each bird family, label into the correct blank box in the phylogenetic tree. So here's Vero. Which one, and Vero looks like it's furthest away from Pitloris. Well, look at Pitloris. And Vero has 1, 2, 3, 4 differences. 4 differences in their DNA. But they already placed those. So which is most closely related to Vero? So let's go ahead and take a look at that. Well, here, Vero and I'm just going to go use a highlighter. Vero and Manuscodia share this C. So that's one similarity. Vero between those. We don't have to worry about Pitloris, right? Because they already put Pitloris on the tree. It's out here, right? So our question is, Vero is here. How close? We need to find the one that has the most similarity. So this is the C for Vero. These two are the same. So of course, this is the same. But this is a difference. So this is a problem. Here, so Mancala has already lost one. Here's differences on all of them compared to Vero. All right. Here are some more similarities. So there's one. Again, these are all similar. Of course, this whole thing is similar here. And these two are similar, right? So we can go ahead and label the similarities, but it looks like the similarities are going to outnumber the differences. To make my life easier, I'm going to say, let me look at the differences between Vero and the other three. So Vero is the same there, but this one's different. Okay, Vero's the same there, but this one's different. Vero's all the same there. Vero's the same there, except these two are different. Vero's the same here, except this one's different. Vero's the same, same. Vero is the same, except this one's different. Vero's the same, except this one's different. Vero's the same, except these two are different to Vero. I'm comparing everything to Vero because I'm looking to see, again, I'm looking to see which one goes here next to Vero, most similar. And the next are all the same. This one here is different from Vero, and this says C, but that's the only one that's an A. It's same. This one's different. It's not an A. This one is not a C. This one's not a G. This one's not a T. This one's not a C. These are all C's, except they're all different for Loris. There's a T. There's an A. This one's a T, but the only one that's a T is that one. So these two have differences as well. So as compared to Vero, this one has 1, 2, 3, 4, 5 differences. 5 differences. And Pidonax has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 differences. And Diphyllodes has 1 difference. 1 difference. Well, which one's most closely related to Vero? What has to be? It has to be Diphyllodes, right? Now in the next box down, this one is much further away. Look at it. This one's further away from Vero than this one. So the next box is the one that's next furthest away from him. So that's the next least amount of differences. Diphyllodes, remember, only had 1 difference. Which one of these differences would bring that family of birds or avians here in this spot? Well, if you look, the Epidonyx, or however you pronounce it, the Donex has 10 differences, but the Manucodia has 5 differences. So Manucodia would probably go here, wouldn't it? And of course, the only one left now is Epidonex. So when you look at how to put these together, we could have went with similarities. Then we would have had to count how many greens we have. But there's so many similarities between the 4 of these that it made it easier to look at the differences and find the one that has the least amount of difference, sort of like we just looked at in the last question. And that would get you 1 point, right? That would get you 1 point. And the other one, there was a couple little arrangement differences. You could have still got a point, but here there's only one possible right answer. If you had put the Epidonyx here, that would be wrong. If you put any other combination in this particular phylogenetic tree with this data, would be incorrect. When we look at this, the scientists have found an allele of a plant gene that may protect plants from some types of insects. To determine whether this allele is present in other plants of the species or is in a recent mutation, they conducted the following experiment. They isolated DNA known to contain the allele from one plant that is unaffected by insects, and that DNA-resistant sample, I think it should be obvious, is listed here since they actually tell you DNA-resistant sample, right? So this is your DNA-resistant sample. Then it says, isolate the DNA that may contain the allele in 3 plants of the same species growing in the same location. So there's 1, 2, and 3, right? In the same location, so you're kind of controlling all the different variables. Treat the resistant DNA sample with the 3 DNA sample and the 3 DNA samples of other plants with the same restriction enzyme and generate smaller segments. So you're breaking this enzyme, you're breaking the DNA up into small bits, and you're running it through a gel and separating. Again, what are we looking for? Just a pattern, a similar pattern. Which one of these has the most similar pattern or different pattern, depending on the question they're asking us? So they're just taking DNA. Remember, DNA is very long. And restriction endonucleases or restriction enzymes are taken from bacteria, and we can use those to cut DNA up into different piece-sized pieces depending on the sequence. So wherever you see T, T, A, C, this one restriction enzyme will cut that here, where another restriction enzyme might be looking for C, C, T, A, and it would make a cut. It would make a cut here, every CCTA. So the sequence, depending on where they cut, it tells you something about how related the two samples are. And then you run them on a gel, and the bigger the piece that's been cut, the further up here it is. The smaller the piece, the further down here it is. The bottom line is that the similar patterns show you how closely related that individual is to the reference, in this case, the DNA resistant DNA sample. All right, so they treat the DNA resistant sample and other samples of plant restriction load. They run a gel electrophoresis DNA segments, analyze the result of gel electrophoresis. Click on the sample that has the same allele as the resistant plant. Which one of these has the same allele, the same DNA sequence based on this data? They didn't sequence it; they just broke it up in a regular pattern. And they asked which one has the same size patterns. Well, is this the same as this? I say no. I think it's clearly no. And I think you can easily see that even though this one's common in most of them, I think it's easy to see that this one's also different from this one. So this is also different. The only question is DNA sample 2. Does DNA sample 2 have it? And if you look, this is the same, 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 same. So this has actually the same allele. And Bob's your uncle, there it is. This is the answer. It's 2, DNA sample 2. Why? Because when I took an enzyme, they cut in between TTAC, in between the A and the C. And sample 1 had a TTAC, T, 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 A, C, 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 A, T, 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 C. Well, this is just an example here. Sample 1, every time there's TTAC, was cut here, was cut TTAC, was cut here, TTAC was cut here, TT, and that was it according to this. So if I cut this sample with an enzyme and a restriction of nuclease, an enzyme that cuts the DNA in these particular sequence, then I'd have three sizes. I'd have a banding pattern that looked like this. One big one, that's this one. This is the big one. There'd be one medium-sized one here. That's this one here. And then one little one that would be all the way down here. Again, the smaller it is, the further down the gel goes. On the other hand, if that same gene for a different species had A, T, T, G, right? T, 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 A, C, C, A, T, 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 C, right? Then you would find that there would be a TTAC here, and they'd cut it, it would cut there, which would only give you two bands. One band that would be this size, and one band that would be this size. So it would be very close together. There'd be one band there would be one band that's smaller, slightly smaller, so it would be a little higher. And there'd be another band that's just very close, but still below. That would be the pattern for this particular example. Then what if you had a third example, like the problem we just had? And this third allele, had A, T, T, C, let's say T, 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 A, G, C, A, T, T, C. Well, that would have just one band. It would just be one band because there is no TTAC. Nowhere in here do you see TTAC. So there would be that would only have one single band in its gel. So if you ran the three gels together, which you'd get if you ran electrophoresis, you'd get one column that would show this banding pattern, one column that would show this banding pattern, right, and then one column that would show this banding pattern, really big piece because it's not broken up at all. So you'd have in a gel electrophoresis that had something that looked like this. These are the banding patterns you'd expect if we did it this way, but they broke it up, and this is what they got. They used a restriction nuclease. They cut a bunch of different places, but everywhere it cut, it's only going to cut between two specific letters in the sequence. Everywhere it cut, wherever it cut this one, you're comparing to these three samples. The most similar one is this one, so these two have the same alleles. These two have something different about them. They don't have as many, or they have more, or what have you. All right, let's go on. All right, this question is how could genetic engineering be used to produce more successful crop in hot, dry climate? Let's look at the options. Non-essential DNA could be removed from the genomes of the crop plants. How would that improve the plant to be able to deal with a dry climate, not as much water? No, that's no good. It doesn't help. I can't think of anything. DNA from plants adapted to dry areas could be added to genomes of crop plants. Well, that makes sense. There's some genes that allow the cactus to survive in a desert, or the tumbleweed that lives in the desert to survive. Well, we could take the gene that lets them survive in that area and put it in our corn or our soy or our tomatoes. Well, that makes sense. So I'm going to go ahead and choose that one. So far, it's the best answer. I'm not sure. I'm going to read all my answers as always. RNA from a variety of non-agricultural plants could be added to genomes of the crop plants. Well, why is that not helpful? Number one, a variety of non-agricultural plants. If you added DNA of these plants to your crop plants, it might help, might not. You'd have to see. It might be helpful, but not RNA. RNA is not inheritable. It's DNA that's inheritable. So that answer is incorrect. mRNA transcripts of genes from dry weather guys can be added. So adding proteins, adding that to the fertilizers, if they could eat resistance to drought, that doesn't make any sense either. So no, that's just crazy. There's only one real answer here, and that's this one. It's the only one that talks about both DNA being added to the genome. And genome, remember, is the DNA of the plant. The other one, this one does mention DNA, but it says non-essential DNA, and it says removed from crop plants. How is that helping? Right. So really, I hope you find that this one is pretty clearly B. And the answer's here, by the way, as you can see. Here's question 8. Scientists study anaerobic fermentation in yeast cells, incubated at different temperatures. Anaerobic, again, just means not using oxygen, and it's using fermentation. It's one of the ways that you can get ATP from sugar. And the amount of carbon dioxide detected at each temperature in 2 trials is shown in the table. As we discussed earlier, I'm going to go and review this, that we talked about photosynthesis, and photosynthesis is when we take CO2 and you add it with water, and you get what you produce is C6H12O6, or sugar, glucose specifically, plus oxygen gas. Now we've talked about this as an aqueous solution. This is a liquid water, and this is a gas. I'm not going to balance the equation, but suffice to say that would be the expectation, the summary reaction that you'd be talking about for photosynthesis. Now, we don't do photosynthesis. That's what plants do and some bacteria do. The rest of us are heterotrophs, and so we're going to take the sugar, that's C6H12O6, and we're going to mix it with oxygen. So we're going to do the reverse reaction. We call it the cellular respiration. And you're going to mix it with oxygen. You're going to get carbon dioxide and water. So we're doing the exact opposite. This happens in a chloroplast. And in the chloroplast, you have the light reactions, light-dependent reactions. And what do? They break down water, H2O, to O2 plus H plus, right? So there's that. They do electron transport, and out of the light reaction goes NADPH and ATP, and they go into the Calvin cycle. And the Calvin cycle captures CO2, right? Captures CO2, and using that enzyme called Rubisco, we talked about, and that Rubisco enzyme captures CO2 in a process that cyclically takes the CO2 and turns it into C6H12O6. All right, that's the ultimate result, is that you're going to produce this molecule of sugar. There's many steps. We could discuss it. It's just not worth our time now. That's what's happening in photosynthesis. Now, in cellular respiration, we're going to add the oxygen to this C6H12O6. We're going to reverse the process. We're going to do that in something called a mitochondria, and you know this. We're going to use an electron transport chain and the resultant processes. We're going to come CO2. It's going to leave. We're going to breathe it out, and in the mitochondria, you're going to take, remember, outside the mitochondria, you're going to do something called glycolysis. And you're going to take glucose C6H12O6. There's a bunch of steps that we discussed. Out is going to come NAD, NADH, and you're going to end up with ATP. The amount we could discuss, you should know there's 2 used, there's 2 come out, and you're going to come at the end after you break down, after you lyse, glycolysis, after you've lysed glucose, broken it down, there's going to be at the end this pyruvate. So this NADH and ATP, NADH is going to go in there. ATP is going to be used. The NADH is going to go in there, and it's going to be used by the electron transport chain to produce more ATP. Remember, we're going to use the oxygen is going to be used in here. Oxygen is going to be in here. It's going to be the last electron acceptor, and what's going to come out is the hydrogens and throughout the hydrogen the electron transfer chain, the hydrogen and the oxygen are going to combine to make water, liquid water. As you can see here, there's liquid water. CO2 is coming out here. That comes out as it goes into the mitochondria. The carbon's gone. In here are the electrons. They get transported from place to place, and we use it to create a hydrogen ion concentration, and you produce oxygen. And that oxygen is the last electron acceptor, and it makes water. Great. Pyruvate is what goes in. That's what goes into the mitochondria. Pyruvate goes into the mitochondria if there's oxygen present and goes and gets broken down, and out comes the CO2. Pyruvate goes in, and CO2 gets broken down into CO2, and the electrons are ripped off and used in the electron transport chain. That's only if there is oxygen. Now, if there is no oxygen, if no oxygen gas, then what happens is that you do fermentation. Now, fermentation is the breaking down of pyruvate again. You get a little ATP, and you have a temporary molecule made. Now, that temporary molecule is in 2 forms. Depends on where you are. It depends on how the molecule depends on where it's being done. In humans, we put it, and in mammals, it's lactic acid, right? In bacteria and fungi, they produce something called alcohol. And that's, in fact, alcohol fermentation is how we make beer and wine and etc. Because we use this sugar. We put it in a bottle or a cask or something. We keep the oxygen out. So the bacteria and the fungus have no choice but to make alcohol, and they make the alcohol for us. We ferment it, and then we distill the alcohol, and we drink the alcohol, or mix it with our drinks, or whatever it is that we want to do. Or use it for fuel, for instance. Lactic acid is what we do in our muscles in our body when we don't have enough oxygen to break down the pyruvate, but we still need some ATP. We get a little bit of ATP out, right? And we produce this lactic acid. It's just that simple as far as that goes. All right, so that's that whole process of respiration and photosynthesis. Let's go ahead and take a look at what the question they're asking us. They're asking us that the amount of carbon dioxide detected in at each temperature in 2 trials is shown. So there's temperature 1, CO2. The more CO2 is produced, right? Because this is anaerobic fermentation. We're producing carbon dioxide. By the way, I should mention that in both alcohol and lactic acid, we're producing, as an output, we're still putting out CO2 gas. And that's why champagne, for instance, is bubbly. That's why when we use yeast, which are a fungus, in bread, you get the bread rises because CO2 is being produced in that anaerobic condition. So we're producing carbon dioxide. Just like we're doing in cellular respiration, in fermentation, you also produce CO2. And they tell you that the amount of carbon dioxide detected at these temperatures in the trial below, so you should be expecting it. Now here's temperature 1, trial 1. The mass of CO2 is listed here. Where is the CO2 working its best? Looks like at about 35 degrees Celsius. You have the most CO2 in trial 1. In trial 2, the most CO2 is still here at about 35 degrees. Now identify the products of fermentation by moving the product labels into the blank box. Well, what's being produced by fermentation, right? We're producing CO2, so we have to be breaking up glucose. So the products of fermentation are going to be, in this case, ethanol, which is, again, alcohol. We had discussed this earlier in the year. It should be a review. And you're not producing glucose. In fact, you're breaking glucose down, right? You're using the pyruvate. You're using pyruvate. So again, that's not a product. And the fermentation products are going to be ethanol and CO2. Well, you should know that. The CO2 you should see because you're actually measuring that. So it's ethanol and CO2. The optimal fermentation temperature, I think that's easy, since we looked at that, is going to be 35 degrees. How do I know that? It's the most amount of product that was produced. Easy peasy. Well, what did you have to do to know this? Well, you had to know what fermentation was. Fermentation is what happens when, and we went through it. We had examples. We looked at we mapped it out. I mean, we did a lot of work on that. And so fermentation is, you produce a little bit of lactic acid in humans, but in bacteria and fungi, you produce CO2. You're certainly not, you're not producing water because you're not using up oxygen. You're not producing oxygen. You're producing CO2. In any respiration, you're going to produce CO2. No matter if it's aerobic or anaerobic, with oxygen or without, you're going to produce CO2. With oxygen, you're going to produce water, but you're not going to make water, right? But without oxygen, you're going to produce CO2 anyways and ethanol. Now, if this were a human or a mammal, it would be CO2 for fermentation and not ethanol, but lactic acid. It's the only change. And again, the temperatures are likely going to be similar. And in humans, it's going to be a different, I mean, it won't be similar temperature, but it'll be a similar result in that there will be an ideal temperature where that's going to give you the best result. Different species have different enzymes, so they're going to work at different, they're going to have a maximum range, and a best range is going to be different for different species because we have different enzymes. All right, so that's question 8. Number 9 is a particular species of mice, a single gene, either big B or little b, and determines its tail length, right? So we know we're going to be dealing with the dominant or recessive, and that's has to do with tail length. A short tail allele is sex-linked and dominant. So let's go ahead and write that down. So short tail, it'd be nice if I use a pen. Short tail is dominant and it's sex-linked. Sex-linked and probably X-linked, right? So it's going to be something like this. So this person here, we know, number 1, we know is X big B. So on her X, she has a big B. Her other X, we're not sure because she has a dominant allele. We have to look at the rest of the pedigree to make our decision. This gentleman here definitely has an X little b. How do I know? He doesn't have the short tail. He has a long tail, right? And he has a Y. Okay. Now they have a daughter that has two X's, and she's long-tailed as well, which means she has to have two little b's. How do I know she has two little b's? Because if she had a big B, she'd have a short tail. So in order to have a long tail, you can only have a little b, either X little b or X little b Y. Now this person here, this male, has a long tail, so he has X big B Y because he has to have a long tail. And he has two boys that he gave a Y to. And the mom in the second generation only has a little b to give. So all her boys are going to be long-tailed because she only has the long-tail gene to give. So now let's look at the question. That's what we have. Individual 6 is a female. So now we know she's a female. So if she's a female, what would be her genotype? We know the dad gave her X big B. We know mom had to give her X little b. So she has to have a short tail. So what is the genotype of individual 6? Well, I just did it. It has to be X big B, X little b, X big B, X little b. It's A. Many different reasons why the rest are wrong. X little b would require her father, number 6's father. Remember, 6 is here. Number 6's father would have to have a little b to give, but we know the dad had a big B because he had a short tail because he has it filled in, see? So this is wrong. X big B Y. Is that possible? Well, no, because they said it's a female. And we know female is XX, so that's not possible. X little by. Well, you could have got the X little b from mom, sure, but the Y will make this what is it, a mouse? This mouse a male, and we know it's a female because they tell you it's a female. So that's wrong. So the only possible answer, even if you couldn't figure all the rest of that out, honestly, the only possible answer is one of these two. You could have got it down to either A or B. And if you're paying attention, you knew that X little b was a long tail. So you didn't even have to do all the work I did here, but hopefully you understood the explanation of how pedigree could help you figure out the genotype, and you should already know that from all the work we've done in class. All right, moving on. Okay, so here, which drawing is representative of a prokaryotic cell? Well, I look at this and I see that this is DNA and there's no nucleus around it. Already I'm thinking this is definitely prokaryotic. So I look at this, I see a cell wall. I see it connected to other cells, other cell walls. I see a central vacuole. I see mitochondria. I see membrane-bound organelles, a Golgi body. I know that's not a prokaryotic cell. This is the same thing. I see mitochondria. I see other structures that are membrane. Here's a nucleus, a nucleolus. Here's a nucleus, nucleolus. So clearly, these are eukaryotic cells. They have membrane-bound organelles. So the only possible answer here is this one here, C. So there's no way it could be any other answer. All right, moving on. Say here in this question 11 is some bacteria have developed a resistance to antibiotics through random mutations. One such mutation allows bacteria to produce enzymes that inactivate antibiotics when they enter the bacteria. Which statement describes how this mutation occurs? The mutation occurs when nucleic acids are replicated. That's certainly possible, right? There could be mistakes in replication. So we have to put that kind of as a question mark because we're not sure it's the best answer, but possibly. Mutation occurs in chloroplasts during photosynthesis. Well, that's no. Come on. Mutations, you're thinking DNA. Mutation occurs in mitochondria, again, during cellular respiration. No. Although that can happen because both chloroplasts and mitochondria have, first of all, let's be clear why that's impossible. I want to point out this says it's a bacteria. I'm remembering that a bacteria is a prokaryote. You use the same spelling. So that means no membrane bound organelles, which I've said about 1,000,000 times this year. So the mutation occurs when nucleic acids are replicated. That's possible. It can't be here in chloroplasts or mitochondria because those are membrane-bound organelles and prokaryotes don't have them. This is happening in the genomic DNA. The mutation occurs in molecules that move across the cell membrane. When molecules move across the cell membrane, that's osmosis or diffusion. That again has nothing really to do with mutations. So it is our first choice was actually the best answer. So it's definitely A. There's more than one way to get this right answer. They discuss proteins produced by information nucleic acids. Mutation occurs in DNA, and DNA replication can lead to that. That's the basic answer, which is why we chose A to begin with. There's really good reasons why these are wrong. And yeah, this is why how they're actually used. That's true, right? But they really should point out this main, really big idea that if you knew bacteria was a prokaryote, you would never have chosen B seed. All right, moving on. Okay, 12. This diagram shows an incomplete crossing of cross-section of the cell membrane. 3 different particles are shown moving across the membrane into or out of the cell. So these guys moving through, this one moving through, this one's going out. Already I'm thinking some stuff before I even look anymore. This is from high. This is going from low to high. That's a star in my brain. Already I'm thinking ATP use is necessary. This is active transport. And the other two, I'm looking at high to low. So I don't really think I need ATP being used at this point in these 2 examples. So I got to go read then what the key is. The key, they're telling me, the green one is oxygen. I know oxygen, what some qualities about oxygen is small. It's about as small as you can get. It's 2 atoms, right? It's 1 oxygen, double bond another oxygen. So it's about as small as you're going to be able to get. It's neutral. It's nonpolar. It's not charged. It's not even just not charged. It's nonpolar. So it says this is going to diffuse easily through the membrane. It doesn't need any help. Glucose, our friend glucose, we keep seeing it over and over again. We know it's big. We know it needs help. We've looked at insulin as an example many times throughout the year. And sodium is charged. It's small. It's actually smaller than oxygen, but it's charged, and so it won't get through easily. So the big one here, glucose, is going to need help, but it's going from high to low, so it doesn't need any ATP. So it needs a channel. So you would have to put the channel protein here. This next one doesn't need anything. It just needs phospholipids. It doesn't need a channel at all. It just needs the phospholipids would go there. And it's phospholipids because it's oxygen, it's nonpolar, and it's small. And then lastly, here you're going from low to high. As I said earlier, you're going from low to high. You need ATP. So the ATP pump is necessary. So that's key. That's that easy. If you know the qualities of oxygen small, then you could easily, and glucose is big. Glucose is something you should know easily. This has a plus charge on it. You know it's charged. So even if you didn't know that oxygen was small and not charged, you know glucose needs a transporter. We've done labs and activities with it. And you know sodium is charged because it tells you a plus. If you see a plus or a minus, that's charged. It can't pass without help. On top of that, you can see that's a yellow square. The yellow square is going from low to high, so you know it needs ATP, so you know it's this one. You know glucose needs a channel. So that only leaves you one to go here. So in multiple ways, this answer has to be placed in this way. All right, let's move on. So here, a biologist is conducting a long-term field study. Notice that there were two types of mice in the valley. One type of mouse had webbed toes, and one type of mouse lacked webbed toes. A dam was built in the valley, turning the land into a wetland. As years passed, all the mice living in the valley were found to have webbed toes. No mice were found to have not webbed toes. So let's draw that out, which is what I like to do and why you get scrap paper during the test. You have two different kinds of mice. Webbed toes, webbed toes, and the other one is not webbed toes. I meant. I guess that's a bad. Let's just say not webbed toed, and the other one is webbed toed. So you have this population of mice. Some have webbed toes. Some don't have webbed toes. Who knows this even number? I don't know. All right. Which statement best explains the observed change in the mouse population? So wait, before we go on, that's the population as it started, but then something happened, some event, some cataclysm, in this case occurring because of humans. We flood the valley. So theoretically, what I imagine is that you used to have this valley, and now there's this water in the valley. Creatures have to be able to swim now. Well, who's going to have the advantage? The mice without webbed toes or mice with webbed toes? Mice with webbed toes can swim really well, or at least better than the not webbed toes, probably. Those that don't have webbed toes, well, they're likely to going to die. Sad but true. So because of the flood, the non-webbed toads die, and what you're left with is the webbed-toed individuals were able to have babies and live and eat and swim around in the water in the flooded valley, while the non-webbed-toed individuals died off or couldn't compete with those that had webbed toes. All right, webbed toes mice are controlled by the environment. Not sure what that even means. The mice grew webbing to move more efficiently in the new environment. Let's be clear, that didn't happen, right? They told you that in the paragraph. They told you that the character already exists, and that's how evolution works. The characters have to be there. There is diversity. There must be diversity present. Without diversity, evolution can't choose. They're all the same. So having diversity, evolution reduces diversity. And the mutation came first, then the natural selection pressure of the valley flooding. So here, diversity, it says the mice grew webbing to move more efficiently in the new environment. That's not true. They had the ones that had the webbing survived. They didn't grow it in order to survive. Web-toed mice survive better and reproduce more than mice without webbed toes. Well, that I think is clear. It's clearly the answer. At least I hope it's clear to you. The increased water levels of the valley increase the mutation rate in the mouse population. Nowhere in this problem set did they tell you that. And webbed toes in mice are controlled by the environment. It's incorrect. I don't even understand what that means. All right. So C is the answer. C is the one that makes the most sense. I would have chosen C. And then if I didn't understand the other three, at least I know I chose my best answer. And I check, and there it is. It is indeed the best. C is the answer. All right, let's move on. Oh, another cladogram. How nice. So you have a cladogram, several members, and phylum Arthropoda. Two new species. Do you know what Arthropoda is? It doesn't matter. But Arthropoda is, they have insects that have exoskeletons, things like insects, shellfish, etc. So the exoskeleton, things with an exoskeleton. Two new species of X and Y have recently been discovered. The table shows traits that these two new species possess. So they have an exoskeleton, they have compound eyes, they have mandibles. The species X doesn't have antennae though. So move X and Y to the proper place in this cladogram. So X has an exoskeleton, it has compound eyes, it has mandibles. So both X and Y have to be on this side, have to be either in one of these three spots because they have all three of these characters. But X does not have antenna where Y does. So X, which has no antenna, cannot be on this side, can't be on this side of the cladogram because anything after this has antenna. Just like anything after these three have it. So you can't put X over here, you can't put Y over here because they have mandibles. So it can't be here. X cannot go past antenna because they don't have antenna. So there has to be here. X has to be here. So you can place your X here for sure. Y species, on the other hand, has antenna. It has antenna, so it's on this side. Head, thorax, and abdomen, it doesn't say anything about those. We don't know if they have them or not. But we can go ahead and put, probably pretty securely put them here, Y species here. Could it be here? Is that a possibility? I don't know. I mean, we don't know. All we know is that it has these four. 1, 2, 3, 4. That we know. We know it has 1, 2, 3, 4. We don't know anything about head, thorax, and abdomen. But for sure it's an XY. All right. So they say you could have put Y here and still got a point. Y could have been here or here because the only thing we know is that they definitely have antenna. So either one of these places would be correct. But X has to go here based on the data we have. All right, very good. Which pair of organelles work together to give the structure and support for animal cells? The cell wall and the cytoplasm. The cytoskeleton and Golgi body. So let's say structure and support. Structure and support. You would think, you see the word skeleton, I hope everybody's keying in on that. You think cell wall, certainly that is going to give you that. Cytoskeleton for sure, and cell membrane, right? And then the cell membrane, structure and support. So when we look at the cell membrane cytoskeleton, you got to remember that the cell membrane is such, and within the cell membrane, you have those microtubules and microfilaments that connect from one side of the cell to the other, and some of them are embedded in the membrane, some of them even go outside the membrane and create some structure on the outside where things are attached. And so you have the organelles, things get moved. Remember the movie of seeing the objects move, the organelles lay within this matrix, this cytoskeleton. So the cell has this ability to move if it moves, or has a shape because of the cytoskeleton, but it's all attached to the cell membrane, so together they work to give structure and function, or structure and support. Now to be clear, the cytoplasm is the liquid part of the cytoskeleton is in the cytoplasm, but they're not attached to it. It's the liquid part of the cell that's in this space here, right? The Golgi complex is that one of the organelles that helps package the proteins for in and out, it going in and out of, for leaving or going to, it helps target where things are going, where proteins are going. And it packages them, sends them off. And then the mitochondria are there, and the other organelles or vacuoles, etc. So when we're looking at Golgi body, that's again I hate that, but let's call it the post office or the packaging center of the cell. The endoplasmic reticulum is where the proteins are made, or the manufacturer factory of the cell. Right? Again, I hate those, but neither one of those have to do with structure or support. The cytoplasm is a liquid part of the cell. Like you can argue so they give structure and support or not, but these two here are the best answer. The best answer when it comes to structure and support. You have to know what your organelles do. Here you have, in most species, scientists have observed that related individuals share about 80% of the same genes. Cheetahs today, however, are known to share 99% of the same genes. When they say genes, they mean alleles. Of course, we all share the same genes if you're the same species by definition, but they also have the same alleles. So cheetahs are incredibly, they lack an immense amount of genetic diversity. You'd expect a lot more diversity. So you think about cheetahs, they're incredibly attuned to run fast, and they're really good at what they do, but they lack diversity. What does that suggest? Are you guys still playing?
Yeah.
How many games are left? Say again? 3 games. All right. Hi, baby. I can't. Hold on a second. Hi, baby. They're finishing up their match. They had to start late, but they're finishing up. They should be done. All right. So, and I'll be ready. I think I'll be ready. You guys will be done in 20 minutes. Is that right? I mean, don't rush it if you have to.
Yeah.
Okay.
Assumably. Sure.
All right. Where are you going? What are you doing?
That's revenge, brother.
All right. Love you, baby. Give me a call when you get here. Bye.
Try to do your best to get her done.
Okay, so when we're talking about cheetahs, how do you get a population? You have to ask yourself, how do you get a population of individuals that have 99% the same? 99% the same. So let's look at the possibilities they've given us. I'll tell you the answer in a minute, but let's look and see what we have. What do we have to choose from? What does a lack of genetic diversity imply about the history of the cheetah population? Well, if they're 99% the same, let's think about what that means. In fact, I changed my mind before we go to the answers. Let's think about that. That means they probably had very similar common ancestors, right? Very recently. So maybe it was a small group. They must have had a small group of individuals that led maybe 2 or 4 individuals. Who knows how many? You could probably do the math and figure it out if you were really good at genetics, and they probably have. But some small group of cheetahs ended up being the parents, the founding members of all cheetahs. So I'm going to use a Bible reference because I think it's something a lot of people have heard of. And if you haven't heard of it, sorry, I don't know. I'm just thinking of what other example I use. But if you think about Noah and the flood, before there were all kinds of people, but then after the flood, right? Theoretically, according to the story, and if you're not Christian, forgive me, or if you're not religious, forgive me. This is not about a religion; it's about the story. So theoretically, before all these individuals existed, then 2 of every individual came on. The only people that survived were Noah and his plus fam, right? Everybody else theoretically died. And so from then on, we're all descended, theoretically, from this small family, from Noah and his family. All the individuals on the planet descended from this small group. Before, there was a lot of diversity. Afterwards, there's very little diversity. Even if it was Noah's family, some friends, either way, there was all these different places and languages, etc. And then afterwards, there's very little diversity. There's probably a lot of people. Over time, populations isolated. They had mutations, and you develop more diversity. But before, there was a lot, and afterwards, there was very little. So maybe there were a lot of different kinds of cheetahs and different alleles in a population, but something happened. Something happened, and wiped out a lot of the cheetahs, and only a small group of individuals. Only a small group of individuals survived and were the parents, the great 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 grandparents of all cheetahs. Very similar to what happened with Noah's flood. So we call this, and you've learned about it in class, but I'll go ahead and say it. We call this a bottleneck. A bottleneck event. Something happened that reduced the diversity in the cheetah population severely, and only allowed a very small percentage of them to survive. And those that did were the small group of genes that all cheetah now share. That's bad news for the cheetah, because unless nothing changes for a long time, and allowing their mutations, so their mutations are slowly coming into being. They don't have enough diversity to deal with a big change in the environment. And of course, global warming is changing their environment drastically. So they got real problems. Cheetahs, I'm not sure, are going to survive the next 30, 40 years, 20, even 20, 30 years. If they lose habitat, they're not going to be able to adapt. They don't have the diversity to be able to adapt. They might, but they might not. Their odds are low. than if they had a higher diversity. We call that a bottlenecking event, right? Something bad happened and killed out a lot of the cheetahs with all their different genetic diversities. Maybe over here you had a fast cheetah, but maybe they ate different things. Maybe they climbed more. Maybe they had a bigger range. Maybe they, who knows what the different kinds of cheetahs, 1A, B, C, and D had. But maybe only B and C's survived, and now you have, or maybe only B's survived. That type of cheetah is the only one that survived, and so now all you got is pretty much B's left, sharing about 99% of their DNA. And again, we're talking alleles. I don't like that they use genes because obviously, I hope it's obvious that a species shares all their genes, just different alleles. So what does the lack of diversity tell you? Cheetahs never experience mutations? Well, that's silly. Everything that lives has mutations. So, in fact, that's why they're not 100% the same, right? If they had no mutations, they'd be 100% the same. They're not clones created by some aliens. And cheetahs have experienced reproductive isolation. Well, that's very true. That could be. It could be. Cheetahs inbred with other large cat species. Interbred with other large cat species. No, because then they would have more diversity, not less. Right? So that's not right. That's not right. Cheetahs share a common ancestor with other large cat species. True or not, that does not help explain why they have such a small diversity, such a high similarity. So they've experienced some kind of reproductive isolation, some kind of event that didn't allow access to the other types of cheetahs, other types of large cats. And so because of that, you only have a small group, in this case I'm saying bees, are the only ones that founded this population of genes. This bottleneck event occurred. What could that event have been? Maybe something came and killed off the A's, C's, and D's. Maybe A's, C's, and D's went off, and maybe there was an earthquake and isolated the cheetahs into one group, and only the B cheetahs were present. One of those genetic drift situations that led to a bottleneck. Genetic isolation led to this. And today, you can see humans choosing this. You could see Hasidic Jews or Amish people choosing to have babies only with people that are very much like them. And so you have this reproductive isolation by choice, right? But the rest of us have a large diversity in our genes, and therefore when this event happens, not if, but when some cataclysm happens or when something comes along and stresses out the population, we'll have enough diversity, hopefully, that some of us will be able to survive. And if that happens, then, and the rest die, then you'll see, or at least they don't produce enough babies, then you see there's going to be a lot of similarities on the other side of that event between the individuals that are left over, much like what happened in this story of Noah's flood. All right, let's move on. All right, so a group of students studied 4 different cell specimens and under microscope recorded information about each cell in this table. So they said, okay, well, cell 1 through 4. Cell walls, yes, no. Cell membrane, yes. Cell chloroplast, no, yes on this one, but no on the rest. DNA nucleus, no on this one. All right, so let's go ahead, before I even look at it, let's go ahead and write down what we know. No nucleus, that's a prokaryote. That is a bacteria or archaebacteria. All right, so we have already identified cell 3. What about having a cell wall with chloroplast? Oh, Well, at that point, chloroplast and a cell wall. That's a plant. Done. So what doesn't, no cell wall, cell membrane, no chloroplast, DNA's in the nucleus. Well, that could be a lot of things. Could be, there's many possibilities here, but let's think about what they could be. They could be animal. They could be fungi. Or they could be proteins. All three of them have, except proteins generally have cell walls, and fungi have cell walls. And since neither one of those have cell walls, it can't be, since they don't have cell walls, we know it's not fungi. And it's likely that both of those are animals. That's probably the case, although there are some proteins that don't have cell walls. Hard to tell. All right, which cell specimen is a prokaryote? Well, that was easy. It's 3. We're done. Didn't have to do all that. But I thought I'd go over that because we don't know what kind of chart they're going to throw at you on Friday. But if you know the qualities of the 3 groups that we've hit on, or 4 groups we've hit on all year long, you should be able to identify that. All right, we're moving on. Okay, function of plant cell organelles. So the stacks of thylakoids, you should already be thinking chloroplast. What's a chloroplast? There's the nucleolus. This is endoplasmic reticulum. Rough or small, it's hard to tell from the picture. This is mitochondria. Again, Golgi body and cell wall. Which one of these is going to modify proteins and transfer them out of the cell? So 2 of them are going to be involved in this. Identify 2 cell structures involved in function described. Please place a structure number into each of the boxes. Do not need to use numbers, all the numbers, and place only one number in each blank box. There's only 2 blank boxes. I obviously can't use all numbers. Modify proteins and transport them out. Well, to make them, they're being made here now. A rough endoplasmic reticulum or ribosomes are free-floating. And the DNA and RNA are necessary to make the mRNA to do the translation that's occurring out here. This central vacuole is here to hold the water and help osmoregulate, perhaps even store some starch. But this Golgi body is here to help package things and move them out of the cell. So that's important, right? Package and transport out of the cell. But of course, in order to be transported, they had to be made. The proteins had to be made in the rough endoplasmic reticulum. If they're made out here, they're never going to get to the Golgi body. So they get they're made and put into vesicles here by the endoplasmic reticulum. So it's 6 and 2 as the answers to this. 6 and 2. 19, number 19. Scientists construct a cladogram. They show you the cladogram here. It's based on evidence collected from DNA analysis. Clusia, the genus is Clusia. These are all related. So these share a common ancestor. These are more closely related than these. And these 3, and this the least the least related to these. The least number of similarities is here. The next least is here. These 2 are very similar to one another. The closest relatives. That's how I would read this cladogram. Which conclusions can be supported from the cladogram? C. Val and C. Rose are the most closely related of the 4 species. That's absolutely not true. We just said Rose and Cylinder are the 2 most closely related. So we know the first one's wrong. Rose and Cylinder are the most closely related. Well, I'm going to say probably this one, but I'm going to read all of them because I want to make sure. C. Cup and C. Val are the most closely related of the 4 species. That's not true. C. Cup is here and C. Val is here. They're not the most closely related. So I'm not going to put that. And C. Cup and C. Sil. C. Cup And see, still, again, not the most closely related. So the most closest, easy one, the most closely related one is, in fact, C. And right on there. So that's correct. Why are they the most closely related? Well, again, they are the ones that we find here, the ones that share the one common ancestor. All the rest of these are related to one another, but further back in evolutionary history. All right,